At the end of the lecture, the student will be able to define the different types of immunity and the mechanism of humoral immunity, to discuss the development of antibodies and their structure and function. The immunity is of two types, the innate immunity and the acquired immunity. The innate immunity is the first line of immune response which relies on the naturally mechanisms which are present already before birth, whereas acquired immunity is the second line of response and it always requires antigen exposure. It is provided by the activation of B and T lymphocytes. This is the diagram showing that the innate immunity is provided by the white blood cells already present within the blood. It is provided by the skin barriers. It is provided by T cells and natural killer cells, whereas acquired immunity is provided by the activation of either B cells or T cells. The types of acquired immunity are humoral immunity and the cell-mediated immunity. The humoral immunity is mediated by the activation of B lymphocytes, which produce antibodies due to the exposure of antigens, whereas cell-mediated immunity is provided by the activation of T cells, which produce cytokines and activate macrophages to deal with antigens. Now the B lymphocytes, they are produced within the bone marrow from the hematopoietic stem cells, which give rise to the common lymphoid progenitor cells. The liver and bone marrow preprocesses the B lymphocytes. In mid-fetal life, the liver is responsible for the preprocessing of the B lymphocytes, whereas later on, this function is provided by the bone marrow. The B lymphocytes have greater diversity than the T lymphocytes in forming the billions of antibodies, which have the function of specificity. It means they are specific for specific antigens. The B cells are activated by the binding of antigen to the receptors which are present on the surface of the B cells, after which there is proliferation of the B lymphocytes. Some stimulated B cells become plasma cells which secrete antibodies, other become long-lived memory cells which can be stimulated at a later time to differentiate into the plasma cells. The development of B lymphocytes occurs after the exposure of antigens. They enlarge and form lymphoblast. The lymphoblast increase their cytoplasm and there is rough endoplasmic reticulum proliferation to form plasmablast. The plasmablast divide once in 10 hours for about 9 divisions, giving in 4 days about 500 cells. They then converted to mature plasma cells. The plasma cells produce gamma globulin antibodies, about 2000 molecules per second for each plasma cell. This is the activation of B lymphocytes. The B lymphocytes enlarge to form the lymphoblast. The lymphoblast further divide and give rise to the plasmablast, and the plasmablast then divide rapidly for about 9 divisions to give the mature plasma cells. This is the step for proliferation of B cells. During formation of B cells, there is the reservoir of B cells which are specific for a specific antigen, and they are reserved as memory B cells, and they are activated due to the exposure of secondary infection. What is the difference between primary and secondary response? The primary response occurs for the first time. It is slow in onset, and there is the antibody formation, which is also the prolonged process. Whereas the secondary response occurs after the second exposure to the same antigen. It is more rapid, more potent, and duration of the forming antibodies is more prolonged as compared to primary response. It means more and more antibodies are formed for the longer period. You can see here in primary response, the main antibodies are the immunoglobulin M, and these immunoglobulin M has 10 binding sites. It means it can bind up to 10 antigens, and which help in eradication of the infection rapidly. In secondary response, there is the great excess of immunoglobulin G, which is the bivalent antibody. It means it can bind up to 2 antigens. What is the nature of the antibodies? These are the gamma globulins having higher molecular weight, that is 1,60,000 up to 9,70,000. They constitute up to 20% of the plasma proteins, and they are composed of 2 heavy and 2 light chains and 2 portions, that is variable portion and constant portion. The variable portion has the function to bind the specific antigen. The variable portion has the antigen binding site, and one antibody can bind one type of antigen. It means that it has specificity for only one type of antigen. This is the constant portion, and this is the variable portion. These are 2 heavy chains and 2 light chains. These are the different types of antibodies: IgG, IgE, IgD, IgM, and IgA. IgG is present in greater amount during the secondary response. IgM is present during the primary response. It has 10 binding sites, whereas IgG is the bivalent 2 binding sites. IgE is present during the allergic processes, allergic reactions. IgD has not very well known functions. IgA is present in mucosal secretions and in tears. These are the different function of different antibodies. How the antibodies act during infection? Either they have the direct action on the invading organisms, or they activate another system of the proteins, which is known as the complement system. The direct action of invading organism occurs by agglutination, precipitation, neutralization, and lysis. What is agglutination? On exposure of antigen, the antibodies bind with the antigen to form the antigen antibody complex, and after forming that complex, there is the clumping of the antigens. This process is known as the agglutination. Precipitation: the antigen antibody complexes form insoluble larger material which form the precipitates of antigen antibody complexes. Neutralization: The antibodies cause the antigen to be ineffective. Its toxicity is neutralized. What is lysis? It is the breakdown of the cell membrane of the antigen. Activation of complement system. What is complement system? It is a defensive mechanism consisting of over 30 proteins. It is produced by the liver and found in the circulating serum. The function of complement system is to kill the microbes in different ways, such as opsonization, inflammation, cytolysis, agglutination, neutralization, and chemotaxis. The complement system can be activated by two pathways, the classic pathway and alternative pathway. The classic pathway is activated by antigen-antibody reaction, which causes activation of specific reactive sites on the constant portion of the antibody. This stimulates the C1 proenzyme and then the cascade of reaction begins. Whereas alternative pathway occurs without the intermediation of an antigen-antibody reaction. It is activated in response to large polysaccharide molecules in the cell membrane of some invading microorganisms. These substances activate and react with complement factors B and D, forming an activation product that further activates the factor C3, after which it has the common pathway with the classical pathway. You can see here the complement system. It has the protein molecules from C1 through C9 and also the protein B and D. So 9, C1 and C through C9 and B and D. The C1 is activated by the classic pathway and B and D, these factors are activated by the alternative pathway without the intermediation of the antigen-antibody reaction. You can see here the role of different activated factors of the complement system. C3b is concerned with the opsonization of the bacteria. What is opsonization? It means that this factor causes the bacteria more susceptible to phagocytosis. Then C3, C4a and C5a. It activates the inflammatory reaction by activating mast cells and basophils, which release certain mediators of inflammation such as heparin and other substances. Histamine. C5a, this protein of the complement system helps in the chemotaxis of white blood cells. What is chemotaxis? It is the process by which the white blood cells are attracted towards the chemical substance which is produced during the inflammatory or either during the infection. Then C8 and C9, when they are activated, they form the another complex C5b 6, 7, 8, 9. This complex helps in the lysis of the cells.